Today we're starting with data sheet 3-12 for your microscopy and staining for your morphological unknown observations and interpretations. We're starting with this group of bacteria here today, and we're excluding this one. I've broken this up into a more blatant chart for you to completely understand what's going on. So let's start up at the top. Step 1, how to split these bacteria up into groups so we can tell them apart. Do a gram stain. So when you follow your gram stain procedures, you will either get gram-positive bacteria that stains purple, or you'll get gram-negative bacteria that stains pink. So you'll either have this group or you'll have this group. Step 2, what can we observe to tell these bacteria apart and split them into groups? Let's observe cell morphology. So if you have a gram-positive purple bacteria and it's rod-shaped, it's probably one of these 3 species. If you have a gram-positive purple coccus bacteria that's circular, it's probably one of these 3 species listed here. If you have a gram-negative pink stain bacteria that's a rod, you're going to have a very easy day with this one species. If you have a gram-positive coccus circle-shaped bacteria, it is this species here. If you have a spiral-shaped or squiggly-shaped gram-positive pink dyed bacteria, it's going to be one of these 2 species right here. So if you don't already have your worksheet completed, let's move on to step 3. How do we split these bacteria up in order to tell them apart by using the tests we've already learned to do in lab? All right, let's zoom in. For those gram-positive purple bacteria that are rod-shaped, you can do an acid-fast stain. If you have an acid-fast positive microorganism, it's going to be Mycobacterium smegmatis. If you have an acid-fast negative blue bacteria, it'll probably be Bacillus subtilis or Corynebacterium xerosis. If you have a gram-positive organism that is coccus in shape or circular, you can break this up by the cell arrangement. If they are clusters, you're probably going to have Staphylococcus epidermidis. If they're in chains and pairs, it's probably going to be Lactococcus lactis. If it's in tetrads with those 4 corners next to each other at 90-degree angles, singles or pairs, it's going to be Micrococcus luteus. If you say, but Katie, these both say pairs, what if I get them confused? Well, we can do another differential test to tell them apart. Or if you have gram-negative pink bacteria that are spiral shaped, you can do a wet mount. So if it's motile, positive, it's going to be Pseudomonas aeruginosa. If it is non-motile, negative and stationary, it's going to be your Shigella flexneri in this exercise. All right, backtracking to the ones you're curious about. For these acid-fast negative blue bacteria that we were talking about earlier that are gram-positive purple with rod-shaped, you can do one of 2 tests, or if you're an overachiever, you can do both tests. You can do the wet mount to find your motile Bacillus subtilis or your non-motile Corynebacterium xerosis, or you can do your endospore stain to get a endospore negative Corynebacterium xerosis or an endospore positive green Bacillus subtilis. If you still can't tell apart your gram-positive purple coccus shaped bacteria, even after looking at their cell arrangement, you can do another test, like the endospore stain, to tell apart your Lactococcus lactis and your Micrococcus luteus. For you see, if you have an endospore positive malachite green spore shown on your slide, it's probably going to be Lactococcus lactis. But if you have no malachite green endospores on your slide, it's probably going to be Micrococcus luteus. And congratulations. Hopefully you now know what you're doing for lab on Thursday. Please take a screenshot of this if you are confused and need to bring it with you to lab.